good morning, good afternoon, and good evening. So we are very fortunate to have three incredible speakers today. We have Dr. Aaron Palmon, who is Professor and Vice President for Research and Development at the Hebrew University of Jerusalem, and also President of Saliva-based. We'll be sharing about the development of the first saliva-based pregnancy test.
So thank you, Sam. So as Sam introduced me, I will talk about saliva lateral flow based for home use. I will begin with introduction, then I will talk about technology and clinical implications, and finally a little summary. I was first familiarized with this project a bit after 2002, when the NIH put a request to the scientific community to develop salivary diagnostic technologies for early disease detection. And when we look on the components, biomarker components of saliva, there are many. My group was focused on the protein level. When we look on saliva protein detections, there are many advantages, but also challenges. And some of them are low biomarker concentrations, masking characteristic that reduce the detection ability of biomarkers in saliva, and inhibitory agents within saliva that block signal visualization with lateral flow. So our aim was to develop a home saliva lateral flow test, beginning with pregnancy, but not only. And our aims were that this test will be chairside, simple to use. Procedure will take up to 15 minutes. It will have similar sensitivity to other tests such as urine, and of course will be affordable. And we began with questioning women about what will they prefer. Would they prefer saliva or urine? And this is before there is any market education about saliva pregnancy test. And not surprisingly for us, about 70% of women preferred saliva over 10% that preferred urine test. When we divided it into age groups, we saw that younger women, about more than 80% preferred saliva test, while older women were a bit indifferent, and only a little bit more than 60% preferred saliva test. So to develop such tests, we had to develop new core technologies, and we can divide them into three aspects. Saliva modifiers, we understood that in order that lateral flow will work with saliva, we should modify saliva, otherwise it will not work. In certain cases, we need to optimize the lateral flow strip for saliva, and of course, develop a special device that will be very user-friendly. This is an example of spiking of beta-HCG in saliva and in urine. When we put negative sample, we can see the control line in saliva and in urine, but no test line, which means no false positive. When we take 10 milliunits of recombinant beta-HCG and add it to saliva, again, we cannot see the test line. However, we can see a faint line in the urine because this is its limit of detection. When we use our technologies and take 1 milliunit, which is 1/10 of the limit of detection in urine, and put it in saliva, we see a very clear band. When then we went into a clinical trials, we took more than 150 women for 1 to 3 days for missed period, and we got a very nice, clear test line, which represent 97% sensitivity. And when we take negative control, we didn't saw almost any test line, which represent 96% specificity. And if you look on these numbers, these are comparable to the high-end urine test. We also developed a COVID-19 detection assay, and what was interesting is that when we used it in saliva only, we got about 70% sensitivity. When we used it as a nasal only, we got about 60%, but when we used it in combination in the same device, we reached more than 90% sensitivity in detecting COVID-19. So how does it work? So this is the device when it is assembled, and when we disassemble it, we see that it is comprised of 4 parts. The first part is the collection handle, where there is a sponge to collect saliva and an indicator that indicates that a minimal amount of saliva is reached. The handle is then twisted into the lab, which is like a shape of a sleeve, where there is our technology. And this technology is adapted for different applications. So it means we are making changes according to the application. Then the saliva leaves the sponge, modified in the lab, and reaches the detector. In the detector, we have the lateral immunoassay strip, which changes the color, and everything is enveloped by a spill protector, which means that no liquid, no saliva will get out of the device, which is important for infectious diseases. This is the enlargement of how the indicator is working. The indicator before saliva is yellow, and when there is enough saliva, it changes its color to deep purple. And this is the sequence: no saliva, little bit of saliva, more saliva, enough saliva, color change. This is how it looks assembled: no sample, negative sample, only control line, positive sample. And these 2 assays were CE approved for self-home use. Another variation of this device is as a collector for other applications, where there is the same handle, the same laboratory, but at the end, instead of a detector, there is a tube that can go to other applications like PCR, or into a dropper, which can go to other lateral flow cassettes. And this is a sample for PCR. We made the device for collection of saliva for COVID-19, where in the lab we put the lysis buffer for the RT-PCR in a dry phase, so saliva pass it and goes to the tube. This is 72 hours, 3 days after collection in room temperature. If there is no processing, you can see the color. If there is processing, it's clear. This is plating it on agar blood plates. No treatment, many colonies, treatment, no colony. And this is the results of the CT, which are similar between immediate use of RT-PCR to 3 days room temperature. And the next question we ask, is it good only for these applications, or it can be used as a generic one for other applications? So this is some hints for that. We tested it for H. pylori, and also with H. pylori, we could detect its presence while we are using our technology. Without our technology, almost no bands. And this is a sample of a clinical one we made on January, a negative person, a positive patient. So we can detect H. pylori also in saliva. This is much easier than the other test in the market. We used it also for the detection of candida. Again, we can see, without, it's difficult to see. Several years ago, we tested it also for malaria. And in malaria, we made a clinical trial in Uganda in 2018. And of course, with blood, it can be seen. With saliva, with treated saliva, in that time technology, we improved from that time. We could already see a faint band, and if we would decide to continue working, I guess this should be improved largely. So to conclude, it is possible to detect HCG and other biomarkers in saliva using lateral flow immunoassay to achieve best detection ability. Both saliva sample and lateral flow immunostrip should be modified, and the technology developed can be adapted for other protein biomarker candidates. And finally, I just want to acknowledge my people. These are my four co-founders, Omer Deutsch with DMD PhD, Guy, Yoav, and Raluca are PhDs. All of them were in my lab and then moved to Salignostics. And to acknowledge our financial support, we have private investors. The NIH RADx program sponsored our COVID-19 project, and the Israeli Innovation Authority gave us several grants for this development. So thank you very much.
Thank you for that, Dr. Palmon. And I see that we have a question that came in through the chat. And if anyone else has a question, please feel free to post it there or use the raised hand feature and we'll unmute you. This first question is, how do you handle the viscous saliva?
Okay, so the trick is in the modification of the saliva. Okay, the saliva we all know is a very variable medium between ourselves morning and evening and between different people. So this was one of the challenges, that we can take saliva of any kind, and through the modification in the lab part, we standardize it. If indeed we would not be able to standardize it, it would not be possible to do such tests. So everything is in the aspect, what I call modifiers. So in the lab, we are doing saliva modifications about the physical properties, the chemical properties. We concentrate it, we move masking effects and so on. And everything should be done in a second when the saliva is going through this set of modifiers. So this is how it works.
Thank you for that. Any other questions for Dr. Palmon? I don't have quite so much of a question as I do a comment, and I think that something as simple as the color change to let someone know when they have the right quantity, I thought was very, very brilliant in the community engagement work that I did back with COVID testing and getting folks to understand, even when they could see through the tube, how much saliva we needed. A color change or some way of like having them be able to independently know that would have been really, really helpful for our team. So
it's a big problem. Okay, what we wanted to gain with this indicator is that it will indicate that you reached the minimal amount needed. Okay, because if you have more, it's okay. But if you didn't reach the minimal amount, it's not working with saliva. Okay, so we worked about a year to develop this indicator that will have this effect.
Thank you, appreciate it.
And we have.
Oh, go ahead, Sam. I think you're saying.
I was going to say we have one more question in the chat. Is your product available for sale in the United States?
No. Now it is only CE approved. The COVID was sold a little bit in Europe. Our problem was that it was too expensive for the European market compared to Chinese product. And the pregnancy test we are currently launching it. I hope that in 2 months it will go to market, beginning in England and in Israel and some other countries, and then we'll see how it goes on. To get feedback from the real life, not just for market experiments.
Sure. And then we have one more question that came through, asking, actually two now, how much saliva is needed for a test is the first question.
Okay. For the pregnancy test, we asked for 450 microliters. For the COVID one, we took a little bit more than 500 microliter. So usually for untrained patient, okay, first time, it will take about 90 seconds. And after 2-3 times, the time is reduced by half. It's a problem. We all think that saliva is very easy to give, but for the untrained patient, it is not. So this is the average times that we found to be useful.
Thank you. Then we have one final question that came through. Is the modifier embedded in the lateral flow?
No. The modifier is within what I'm calling the lab, the sleeve that you saw. Within the sleeve, there is a part which is a set of filters, different filters with different compositions. As I said, we optimize, although there is we have like a toolbox of components that we can put in this lab. We optimize the tools according to the application. So the lab for the COVID will be a bit different from the lab for the pregnancy, okay? And in this way, we get optimal results.
Great. Thank you very much. With that, we can close out the question and answer for at least this speaker. Very much appreciative of you and your presentation, Dr. Palmon. Sam, back over to you.
Sure. Well, thank you so much. And everyone, if you have any other questions for Dr. Palmon regarding his presentation, feel free to put those in the chat and we can revisit them at the end of the webinar. So we will be moving on to Dr. Daniel Olsen, who is the Associate Professor of Pediatric Infectious Diseases, and his colleague Karen Arias. They will be sharing on nasopharyngeal swabs versus saliva testing for the detection of SARS-CoV-2 in Guatemala. So over to you all.
All right. Well, thanks so much. And thanks for the invitation. We also appreciate Saliva Direct being helpful in helping us set up our saliva testing in Guatemala. And so I think, as we heard eloquently from Dr. Palmon, that saliva has been a really useful test and increasingly used for many different applications. Lower and middle income countries also have, there's a need for increased saliva use in lower and middle income countries as well. And so I'm going to pass this over to Karen Arias, who is the lab director at our site in rural Guatemala. And she's going to present her thesis project, which is comparing saliva to nasopharyngeal swabs for detection of SARS-CoV-2. And then we'll also spend a little time talking about how saliva has been a useful sample type for the several research projects that we're carrying out at our site in rural Guatemala. And so, Karen, I will leave it to you if you want to share your slides and take it from here.
Okay, thank you so much, Dan. Like Dan says, I will present our preliminary data about our study. That is a small cohort about another huge study that we are doing right now at our site. Okay. Obviously, all knows that COVID-19 test plays a critical role about fight against the virus. IDSA recommends RT-PCR testing for SARS-CoV-2 with nasopharyngeal swabs like a gold standard and reference as standards for all this kind of tests. Nasopharyngeal specimens collection causes discomfort and generation of droplets, which are hazardous for our healthcare workers collecting these specimens at the site. And also saliva presents as an attractive alternative for all the detection for SARS-CoV-2 with a report sensitivity of 82% compared to 84.8% for nasopharyngeal samples, respectively. Our objective for this study is the evaluation for the performance of saliva samples compared to the nasopharyngeal swab in the detection for SARS-CoV-2 on the Roche cobas Liat platform at our research site in rural Guatemala. Our study comprehends a huge study right now in our site is agri study that enrolled about June 2020 until this day, 2,107 farm workers that are enrolled right now for this study. This study is influenza like that we weekly worksite visits have for all these workers and have sentinel visits to find out who are symptomatic of these farm workers. And also we research flu, RSV, SARS-CoV-2 with PCRs in Roche platform. And for just to know, COVID-19 vaccine for these farm workers are about 81% workers received 2 plus the 2 doses for vaccine for COVID. And for this kind of workers, 42.7% receive a booster for Pfizer or Moderna kind of vaccines. And to this population, 45.9% has anti-N IgG antibodies for COVID-19 until 2020. That is a presumptive results. For these cohort characteristics, we show about 1970 workers that are symptomatic for agri study. The mean age was 30.8 years. The majority are male, about 77%. And the maximum duration for symptoms in this kind of workers was about 5 days, is the most common. And the most common symptoms for these workers was cough, about 15%, fever and cough, fever, and combination for fever, cough, and difficult from breathing. And the less common was difficult from breathing. Our methodology is very simple. For agri study, we take some samples and do some studies and search about some symptoms common for these workers, that is fever, cough, difficult from breathing. And then we take 2 kinds of samples, nasopharyngeal swabs that take the nurses at his homes and replace all the swabs in UTM immediately. And saliva was taken in conical tubes. Both are taken in coolers, obviously with cold chain transportation for the lab. And then in less than 30 minutes, we do some saliva dilutions. In the case of saliva, 1 to 2, in saline solution, about 0.85%. This is for reduce our viscosity for the samples. And then place the sample, like nasopharyngeal or saliva, in our test. That is very simple, about less than 100 microliters, and replacing the machine for cobas Liat. This is an example for our machine. We have 4 machines for this study run at the time. And we have a result, PCR result, point of care, in about 20 minutes. And it's very simple way to have a result that we can share with the patient at the time, and then take some recommendations and follow him for the illness. Our result shows about 97 patients right now that we have a very good sensitivity and specificity for our study and comparison. And we have a great results because, as you see, we don't have this discordation about these results. It's very low cases that we have discrepancies. As we can show you right now in this table, for the subjects that have nasopharyngeal negative result and positive result, we just have 3 patients. All are male, and our CT values are very, very high. And the most common symptom was cough and fever. don't show in this table, but the symptom was about 5 days, sorry. So it's very common that we have a negative nasopharyngeal swab, but we have a positive saliva test. If we have more days for symptoms, it's more easily that we have results for saliva positive than nasopharyngeal swab. But in the second part of the table, we show you 5 subjects that have nasopharyngeal positive result and negative result for saliva, but was less days for symptoms that present all these subjects. So for less day for symptoms is more usual that we have nasopharyngeal positive results. This is the most common symptoms that we show in this table. So right now, I'm gonna share you this other study, is AgriCAS study. It was a part of Agri study that at the beginning started like a influenza study. Then we have the pandemic, and appears another question about how is the saliva can be a very good option and tool for take the samples for this kind of test, right? Because all we know that a nasopharyngeal sample is difficult for the patient to take at the site. So saliva is more comfortable for them and more easily for us like health workers that we are working all the time with this kind of test. So our CAS study starts in August of 2021. This study enrolled households for AgriCAS as objects. It's around 70 homes right now. And obviously have a surveillance that we weekly have visits and samples all the time for saliva. It's just saliva sample for this study. And we use PCR and NGS that we are progressing and start to improve right now in our site for COVID too, and also we are trying to implement influenza too. For this study, we are visiting more than 33,000 visits, and we have 60,000 saliva samples in our site. For all these visits, we have 88% visits positive for COVID in households until these days. So our conclusion, preliminary conclusions, because we are in surveillance right now, is that shows good agreement between nasopharyngeal and saliva samples and detection of SARS-CoV-2 on Roche cobas Liat performance platforms. In our findings, we support the collection of saliva samples in the detection of SARS-CoV-2, which is most acceptable for the patient and safer for us to take the sample. And also, as you see in the AgriCAS study, saliva is a preferred sample type for the community. It's more acceptable for all the patients that we are enrolling and surveillance around the study right now. Okay, well, as you can see, it's a lot of people behind our study. We have a lot of people of the Center of Global Health, CSU in Colorado, CUCOM. Also, we have on our side right now is in a small part of Guatemala, a rural area. It's all of these people that you see in this bus. Also, we have the help of the University of Del Valle in Guatemala City that can help us in all the time. CDC persons and all these people is behind our study, and I'm very glad to be part of this huge and very, very good team. Thank you so much.
All right. Well, thanks so much, Karen. I think we have a few minutes for questions too. But just to kind of reiterate the second point is that, this household transmission study we visit these households twice every single week. We collect a saliva sample every week. And part of the reason for that is if we identify a COVID case, we want to go back retrospectively to identify a pre-symptomatic or an asymptomatic case that brings COVID into the home. And if we didn't have saliva, this study wouldn't work. This community, I think similar to many other communities in the world, they prefer saliva. They do not like nasopharyngeal swabs or nasal swabs for that matter. And having just participated in a study with my own family in the last couple of weeks, I can attest to that. And so, yeah, I mean, saliva for this, this is great, and it's really made that study possible. So it's been a really kind of interesting experience for the site, and it's working. I think one of the problems is what is a gold standard. You compare saliva and nasopharyngeal swabs, but we've also had plenty of false negative NP swabs as well. And so for us, having both samples has actually been helpful for some of these studies. But I'll stop talking, I guess, and we can take some questions if there are any.
Thank you. I see a comment in the chat of excellent work and presentation. If anyone has, so thank you for that. If anyone has a question, they're more than welcome to post it in the chat or use the raised hands feature in the reaction and we can unmute you. Perhaps while folks are thinking of their questions, actually, Karen, I had a curiosity that relates to the last slide. Are you utilizing community coordinators and things like that as a part of this project? I assume that that's part of your process for making sure that you're being inclusive of those folks you're trying to help serve.
Yes, obviously, our study have a coordinator in the site. Obviously, our PIs are Dan and also Doctora Mireya Calvimontes that is in Guatemala City. So in our site, we have obviously continue meetings and reports that we are also in a lot of communication with all these people, right? Because never working alone. We are a great team.
Absolutely. And you must have a tremendous amount of trust to be welcomed back week after week to be able to do these samples and get those paired swabs, even if nasal swabs aren't the preference.
Yes, and there is a lot of sample because as you see, we are taking about a lot of samples, maybe 5,000 or 5,000 of samples saliva just for agriculture study. It is a part of agri study and also the comparison is a lot of samples, but we are very comfortable to work about that.
Beautiful. Thank you.
If I could just add to that, I think, one of the things that's been really useful is being able to give results back to participants immediately. I think many research studies that just ship their samples off elsewhere struggle with retention of participants because, what's in it for them. But if you can carry out a point of care PCR test in like the resource limited area using a preferred sample type, that's a great benefit to the participants too. And it helps with the study, so you can retain your participants too.
Yeah. Hopefully you're also seeing it building trust for maybe other additional public health services and care and getting folks coordinated in on getting access to that too.
Right.
Yeah.
Well, thank you so much. Really appreciate the time. And if anyone thinks of an additional question they want to post in the chat, we'll be happy to jump in on those and take those at the end. And now I'll turn it back over to Sam to introduce our third and final speaker for today.
But now we will have Dr. Dinglasan present on revealing the unseen towards the development of saliva-based point of need rapid tests to quantify the asymptomatic pathogen reservoir. So go right ahead whenever you're ready.
Great. Thank you so much for having me here. I'm really glad that Professor Palmon actually introduced malaria already. So I'll be focusing on malaria, and let's see if we can get this thing going. So as you could imagine, the pandemic had a massive impact on our ability to continue our malaria control and elimination efforts across the globe, and everything has really been stalled as a result. So as of the 2022, so this is a report for 2021, malaria has been stalled for quite a while, actually. It started stalling around 2016, 2017. And as a result of the pandemic, we are off track by about 48% for our anticipated Millennium Development Goals. So clearly, as you can see from this graph, this represents a reversal of gains that started, back in 2010, and it's these massive gains have pretty much been lost. Good news is we are plateauing. So last year, the World Malaria Day theme, which is in April 25, was harness innovation to reduce the malaria disease burden and save lives. And I thought it was an appropriate goal for today's presentation. So that's really the theme underlying my presentation today. So for those of you who don't know what malaria is a protozoan parasite that's transmitted by female Anopheles mosquitoes all throughout the globe. The heaviest burden for malaria mortality and morbidity and mortality is in sub-Saharan Africa, with several of the countries that I work in, such as Nigeria and the Democratic Republic of Congo, representing close to 45% of the entire malaria burden. So the mosquito injects the parasite into the skin, passes through the skin, into the blood, goes in the liver. And this area here is 7 days or so, and then it breaks out of the liver into the bloodstream and causes disease. A subset of these parasites become sexual stage parasites, which are the only stage that can be picked up by the mosquito, and then allows for the transmission of the parasite. So here I've kind of outlined, for those who are not familiar, this is subclinical in the liver, subclinical in when you have gametocytes and very low asexual parasitemia, and this is the clinical manifestation. And shown here on the right is basically just a general idea of the number of parasites that would be in the clinic is around here. And this is what we would call, on both flanking sides of this curve, the subclinical asymptomatic reservoir. So what do we do in my lab is we try to innovate to end malaria, and we use systems biology in this process. What is systems biology? Well, it's an iterative process wherein you combine biological research with new technological advances in assay development, new computational approaches that takes all of this big data and tries and refine our hypotheses on the biological front. So this could, for most people in the basic sciences, this can go from the cell to the tissue to organism. Our organism could be a mosquito or a human, and then also the ecology of the disease. We take it a step further and we talk about data all the way to intervention. We call it systems translational biology. And again, as you can see in the bottom, the goal for biology is hopefully to make an impact on disease pathogenesis through these new interventions. So here's an example to kind of introduce what we're doing. The basic premise that we had was the parasite can make these sexual stages, male and female gametocytes, shown here, the female and the male. And this sexual dimorphism in a unicellular organism is amazing as a question, but can we really study it? So we had to develop these tailored workflows in the mass spectrometry space. We developed new multi-search engines, so computational biology. We had to come up with new assays because, again, it is a very difficult system to study. And we came up with new computational space. And then what happened? So we published on it, and we said, hey, we have these male and female gametocyte biomarkers now that are secreted and expressed on the surface. And that's what you can actually see here, an antibody against the female that is not, that is very specific to the female. So that led to this question of, can we make a diagnostic with this knowledge? And this is work that was published not too long ago in Science Translational Medicine. So what were the considerations for our target product profile based on the biology is that current rapid tests are based on blood. And for falciparum, Plasmodium falciparum, which is the main parasite species that causes the majority of severe malaria and death by malaria, is on this HRP2 protein, histidine-rich protein 2. Well, the problem is in South America and in parts now in the Horn of Africa, we have seen gene deletion mutants for HRP2 and 3. And here in this model by our collaborator, Oliver Watson at Imperial College, he has modeled what an introduction or expansion of this HRP2/3 gene deletion mutant means for sub-Saharan Africa. So this really goes into this question of gene essentiality for this parasite. For folks who don't know, HRP2 wasn't designed to be a diagnostic biomarker. It was discovered in Hopkins, where I was previously a faculty member, and it was just like, oh, this is present. And so from there, Becton Dickinson kind of took it over, and we are here today with HRP2 being a main biomarker. The problem is, as I said, clinical disease is just the tip of the iceberg, and the majority of malaria we feel is subclinical. And this is the reason why we're also seeing this plateau. So from 2010 to 2016, when we had these massive gains in malaria elimination and control, what we started to see was, well, if we cannot see the asymptomatic reservoir, we will continue to have malaria. And again, RDTs were not designed to be used to identify asymptomatic reservoirs. They're designed to be used in a clinic where there is clinical malaria, lots of parasites, lots of protein biomarkers in the blood. Again, it's a blood-based test. And so if you have this focus, and here's the RDT, and microscopy is becoming a hard to find expertise in the developing world, we're left with PCR. And so the subclinical reservoir is unseen. One of the things I wanted to note was gametocytes can exist in the absence of these asexual parasites that cause disease, or in the presence of very submicroscopic parasitemia. So even really expert microscopists cannot see some of these parasites. Gametocytes can be there. So why saliva? So it's been said saliva is a spitting image of your health. The two preceding presenters have already talked about its utility for different pathogens. And we have this female gametocyte biomarker that we had discovered and published. It's called Plasmodium sexual stage protein 17. And we hypothesized that these proteins, just like Dr. Palmon had shown, parasite proteins in the blood can end up in the serum transudate and then be incorporated into whole saliva. And so we hypothesized that there are two mechanisms in place: cell lysis, so this could be also spleen destruction of red cells. It could also just be natural cell death as these red cells age, as well as the parasites egressing from the red cell. All of these proteins can enter into the serum transudate. So what is PSSP17? I mentioned it already before. It's a secreted protein from female gametocytes. This is the wet test that we had developed and published in 2019. It uses a lateral flow. And here we were using a saliva collection device. I think Paul Slowey's in the call, and to collect saliva. And this is the wet test. Again, a wet test is not a commercial product, right? But we wanted to see just how much of this protein can be detected. And I don't know if you can see it or not, but 1 nanogram per ml of PSSP17 was detectable. We also took this from the lab into the field, and we've collected up to 1 ml using this collection device. We only require 10 microliters of saliva in the end. Again, we're not detecting HRP2. We're looking at PSSP17, and in 393 individuals, we were able to detect PSSP17 in both subclinical. So these are children playing football, soccer for Americans in the field. And then we did it in 3 different countries because malaria can be quite local. And so we also did this in the clinic in Sierra Leone, and the test works really well. So what are the antibodies seeing? So this is PSSP17 in this ribbon model for the protein. Here's the N-terminus. And so we have a capture monoclonal antibody and a detection monoclonal antibody that we have developed. However, one of the biggest costs for lateral flow tests for malaria has been these antibodies. And we run the risk of losing these monoclonal antibodies if we still use hybridoma technologies. So we did RNA-seq and sequenced the full heavy and light chains, variable heavy and light chains of our monoclonal antibodies, and and put them in an expression vector to make recombinant monoclonal antibodies. So now we don't need a freezer. We just need basically we can have this DNA in a Wattman card in my office in perpetuity. We transformed non-adherent Chinese hamster ovary cells and did high yield expression for these recombinant human chimeric monoclonal antibodies, because we found that in developing world, many of the children have an anti-mouse response. So you can actually have some false positive results on your lateral flow. So we had to partially humanize our monoclonal antibodies. So this test is called saliva-based malaria asymptomatic and asexual rapid test 1, or SMART. We have since moved forward into just the collection of saliva to reduce costs. As Dr. Palmon had done, we have the option of taking from the saliva collection cup and putting it into a solution to allow for stability for downstream use on the proteomic side. And then we can also use it for a lateral flow. We use the term point of need because we're trying to break past the current national malaria control programs, which is very clinical based, and we want to move into the point of need. And that means mom, that means the pharmacist, that means your teacher, and so forth. And so this is something that we have been pushing very much so in our field. So this is how the commercial test looks like that we have developed. You will notice that I'm very disappointed in it because we didn't really achieve the same level of sensitivity as our wet test. But this is also expected because whenever you go from lab to scale up, a lot of things can go awry because you really want this stable test, you know, at RT being 33 degrees to 36 degrees, depending where you live. And we wanted to have this stability. And so we did lose a little bit of our sensitivity. If you put your face close enough to your screen, maybe 4 inches from it, you can't see that. You can see that line, right? Daniel, yes, yeah, you can. You're not putting it there. So you can see it, but it's a plus minus. So definitely we want to have further optimization of our dry test. Our goal is 5-fold sensitivity just to hit the lower nanogram per ml. We have already collected 1,600 saliva samples per site this year. We're doing another 1,600 next year, another 1,600 the following year to build up our total saliva collection. One of the things that's really nice about saliva is, especially if you can stabilize it, you don't have to do it at the time of collection. Obviously, fresh is best, but we can also do it unpreserved. And then we also have this nagging question, how much PSSP17 can we actually quantify in saliva? You know, it's 32 nanograms per ml, and this 5-fold increase in sensitivity, does it really matter? When in natural asymptomatic infections, we could actually be in this level. And so we already have a test that's ready to go. So we also had to ask ourselves gene essentiality. I had mentioned before that HRP2 gene deletion mutants suggest it's not required for the parasite. So we examined 9,000 parasite sequences and for the PSSP17 locus, and we could not see any gene deletion mutants. What you see here in light blue is basically some variants, predominant variants across the globe. And thankfully, that variant is just this amino acid change here. Arginine, so the R194 to lysine position does not sit where our monoclonal antibodies sit. So we're in good shape there. However, let's knock out this parasite protein. So we did that through homologous recombination, taking advantage of homologous recombination and CRISPR-based technologies. We show here that we can actually knock out the gene, identify 2 independently derived clones, and immediately we start to see phenotypic changes. So the number of gametocytes that are developed is lower. The number of fully mature gametocytes, which are stage 5 here, are much lower. They're high in the wild type, but they're much lower in the deletion mutants. And then the male-female ratio, which is normally 1 male to 4 females, is now closer to 1 to 1 or 1 to 1.5 parasites. So this actually changes fundamentally the biology. So total gametocyte output is reduced, no stage 5s, male-female ratio is altered. And then it doesn't transmit to the mosquitoes very well. What are these guys then? These are some breakthrough parasites. So now we're finishing up the experiments, going all the way to the sporozoites to determine whether or not these oocysts in the mosquito are actually producing sporozoites. Our gut feeling is no. So the last 2 slides are summary. This is our TPP for SMART1, non-HRP2, an essential protein biomarker, non-invasive saliva is acceptable. It's in review right now, our paper from the DRC. It's immunogenic. and we can use it for multiple things, including research. So this is in prep right now, but we're pretty confident that this parasite, should it develop a null mutation, cannot be transmitted. So lastly, for those in the call, this is the practical aspects. Our goal is cost per test, 25 cents per test, which matches the current RDT. We are allowing for up to 100 microliters of saliva. Right now we are under 15 minutes, hands-on time 1 minute, and we're pretty excited. But also I still want us to hit that lower level of sensitivity before we start going into the field. So that's where we are, and lots of folks, lots of funding to give thanks for. Thank you.
Thank you for that. Really appreciate your presentation. And we already have many questions that came through in the chat. First, being interesting work, is it a gold nanoparticle-based detection is the first question.
No, it's not. And I should have said that. Sorry. No, it's a europium chelate. So it's europium chelate is very, very interesting because you just need a simple UV light, similar to those of you who travel outside the United States, to get your ID, and they use a UV light at TSA checkpoints, same light. And it allows this quick excitation of the europium chelate that allows us to have that level of detection that is easier to see. And we can actually capture this by cell phone already. So no, it's fluorescence detection.
Fascinating. Thank you. Then over in the chat, there's a number of other sub questions to that question. So maybe I'll just direct your attention over that way as I read them. The next one is, how specific is your POC, cross-reaction unspecified signals, unspecific signals?
In terms of, we haven't had the chance to do the sensitivity specificity tests for the commercial tests yet. However, in terms of the wet tests, it's greater than 95% for the specificity. These are monoclonal antibodies, so they do not cross-react with anything else. They're so specific even to the stage, and so it doesn't do anything like that. Our biggest worry was the anti-mouse response that is in the saliva of children. So you get these false heterocomplexes on your test line that is basically your capture antibody and your detection antibody being detected by an anti-mouse. And that has been our biggest problem for false positivity.
Interesting. Thank you for that. And then the last sub question to those is, what about the saliva viscosity, which can be critical, very critical for flow in a lateral flow?
Yeah, so we tested that initially through the lollipop, and which actually gives a very clean expressed saliva, but we found it wasn't needed for our study. Now, why I actually like Dr. Palmon's stuff, so maybe we should talk about it. But yeah, I think there's a lot of variability in saliva. So the more you collect, you can actually have some clear areas versus non-clear areas. But it seems to work without. So we're in pretty good shape in that regard. There are added steps that you can do at the site of use. We're trying to avoid them for processing, but after hearing Dr. Palmon's talk, I might probably need to revisit some of that.
Glad that we can help make connections that amongst our panelists as well as with our guests. Very much appreciate that. Any other questions either for the last presentation or for any of our other speakers? Please feel free to post them in the chat, or again, use the raised hand feature. Well, we certainly are very, very appreciative of all of our speakers joining us today, and for our guests taking the time to join us as well.